It is a great pleasure to present our work published in iMeta. Herein, we will introduce our team's study entitled Single-Cell and Spatial Transcriptomics Reveals Potential Molecular Mechanisms of Abelmoschus manihot L. Medic in Treating Diabetic Kidney Disease. Global prevalence of diabetes has risen steadily over the years, with the International Diabetes Federation projecting over 700 million cases by 2045. Diabetes frequently induces microvascular complications, with 30% to 40% of patients developing DKD. Currently, no effective therapies fully prevent DKD progression to end-stage renal disease, imposing substantial health and economic burdens. The pathogenesis and therapeutic mechanisms remain incompletely understood. To address this, we employ Spatial Transcriptomics Nature Methods 2020 Method of the Year and Single-Cell RNA Sequencing scRNA-seq, Nature's 2019 Science Method of the Year to investigate mitochondrial homeostasis, immune infiltration, and therapeutic mechanisms of irbesartan and TFA in DKD. First, this study evaluated the alleviating effects of total flavonoids of Abelmoschus manihot on classic pathological features of DKD, including glomerulosclerosis, mesangial matrix expansion, collagen deposition, Kimmel-Steele-Wilson nodules, glomerular basement membrane thickening, and excessive inflammatory cell infiltration. Through cell-bin partitioning and TD mapping, a total of 15 cell types were initially identified, encompassing renal tubular cells, loop of Henle cells, distal convoluted tubule cells, podocytes, T lymphocytes, macrophages, and others, which are distributed across 3 tissue components, renal tubules, glomeruli, and immune cells. After rigorous cell annotation and cell marker validation, we further performed transcriptional regulatory network analysis. The results showed that the ligand receptor transcription factor target gene regulatory network of Huangkui capsule in the treatment of diabetic kidney disease, which we previously published in Phytomedicine, was recapitulated in the spatial transcript data. Similar to HKC, total flavonoids of Abelmoschus manihot alleviates DKD by regulating receptors including TGFBR1, IP5, and ICA3, acting on transcription factors Junb, Jun, and STAT1, and modulating pathways such as immune infiltration and fibrosis. In collecting duct principal cells, endothelial cells, macrophages, parietal epithelial cells, and smooth muscle cells, both the STAT1 regulin and its corresponding gene expression were negatively regulated by IRB and TFA. Spatial analysis results demonstrated that the transcription factor STAT1 specifically exhibited a dense, high expression pattern in CPC of DKD samples and colocalized with immune cells, whereas the expression intensity was low in other groups. The target genes of this regulin displayed distinct functional specificity. Functional enrichment analysis of the top 50 target genes most strongly co-expressed with the transcription factor across different cell types revealed that, in C, the target genes of the STAT1 regulin were mainly enriched in pathways related to peptide antigen binding, TAP binding, and TAP complex binding, while in MAC, the target genes of this regulin were enriched in immune-related pathways such as integrin binding, chemokine activity, and cytokine activity. Immunofluorescence staining results showed that alpha-smooth muscle, a key marker of renal fibrosis, exhibited the highest signal intensity in the DKD group, whereas the fluorescence intensity was significantly reduced in the control, IRB, and TFA groups. To facilitate the dissemination of the findings and hypotheses from this study, we have developed an interactive web application based on the spatial transcriptomic data presented herein. This application offers functionalities including gene expression visualization, cell type distribution mapping, pathway scoring, and metadata browsing, enabling researchers in related fields rapidly explore DKD-related data without the need for complex data processing procedures. The main findings of this study are briefly summarized as follows. 1. TFA exerts a therapeutic effect on the pathological progression of DKD comparable to that of both IRB and HKC. 2. Both TFA and IRB can effectively reduce the level of immune cell infiltration in DKD mice. 3. 7 major flavonoid components in TFA can the TGFBR1 pathway and IP5 STAT1 pathway, thereby treating DKD and alleviating key biological processes involved in DKD progression, including fibrosis, lipid metabolism disorders, and renal tubular injury. 4. We welcome researchers to access the DKD-related pathways of interest through our online website.